tumoral cells. Okay. But in benign tumors, this proliferation is localized, it's only in one side. But in the malignancy, we have invasion and metastasis. You know, invasion and metastasis. What's the difference of invasion and metastasis?
Metastasis is over from one organ to another.
To other side, to other parts of the body.
And
But
like
invasion
this one, localized.
Is localized, yes, exactly. Invasion means extension of tumoral cells to the surrounding tissue. Okay, but metastasis is the spread of tumor to the other part of the body that are far from the primary tumor. Okay, for example, about the kidney tumor. Kidney tumor we call them renal cell carcinoma. Okay, why we call them carcinoma? You know about the suffix of
Oma
the
mean
tumor? Yes. Oma is for
malignant.
Oma.
No, sorry, benign.
Oma is for benign. Yes.
Coma for malignant.
What?
Sarcoma for
Sarcoma and or carcinoma. Both are for malignant. For malignant epithelial tumor, we say carcinoma because they are epithelial. And about the mesenchymal tumors, for example, the tumors of bone, for example, muscles and other mesenchymal tissue, we call them sarcoma. Okay. Now when we want to speak about a renal cell tumor, that they are tumors from the epithelial origin, we call them carcinoma. For example, renal cell carcinoma. Okay, a renal cell carcinoma can invade to the surrounding fatty tissue, or sometimes can invade directly to the adrenal gland. You know that adrenal gland is in the superior aspect of kidney. This is invasion. And sometimes we have renal cell carcinoma, for example, in brain, or in bone, or any parts of the body. Okay, this is metastasis. So the invasion and metastasis both are for malignant tumor. You know that the heart of malignancy is mutation, and we say many things about many types of mutations in cancerous cells. Okay, who can name two important mutations, two important genes that their mutation is very important for carcinogenesis?
Like RB gene. Like RB gene.
RB gene, yes,
TNF,
exactly.
TNF 55, 53.
TP53,
Yeah.
yes, TP53 or P53. As you said, P53 and RB genes are two genes that their mutation are very, very important for carcinogenesis and in the malignant tumors. Today we want to speak about some specific features of the malignant cells, some hallmarks of the malignant cells. Today I think we can learn about three of them, that they are reprogramming energy metabolism or altered energy metabolism in tumoral cells, evasion of cell death or evasion of apoptosis, and also the limitless cell proliferation or immortality of the cancerous tumoral cells. Okay. About altered cellular metabolism. Reprogramming very simple way. We know that there are two pathways for producing energy in the cells, in normal cells. Okay, we have one pathway that we call it oxidative or mitochondrial phosphorylation. And the other pathway that we call it glycolysis. We know that in this pathway, the cells can produce about 36 molecules of ATP per each molecules of glucose. Okay. And in this pathway, the cells can produce only 2 molecules of ATP per each molecules of glucose. So, in your opinion, which way is better? Which pathway is better?
Mitochondria. What do you mean? What's the question?
I said that there are 2 pathways for producing energy in the cells, in the normal cells. We have mitochondrial phosphorylation and we have glycolysis. Both of them are present in normal cells. In mitochondrial phosphorylation, the cells can produce 36 molecules of ATP, but in this pathway, in the glycolysis, they can produce only 2 molecules of ATP.
Okay.
And my question is that which way is better?
Mitochondrial phosphorylation.
Mitochondrial phosphorylation. Yes, we think that mitochondrial phosphorylation is better because they can produce more energy. But the malignant cells prefer the other way. They prefer the glycolysis pathway. And this is very important, and we should find why they prefer this pathway. Okay. As I said before, the cancer cells demonstrate a distinctive form of cellular metabolism. They use the glycolysis pathway, and in this way they need a high level of glucose and glutamine uptake. They have very high level of uptake and absorbing of glucose, and they show increased conversion of glucose to lactate or fermentation via the glycolytic pathway. It means that the malignant cells, the tumoral cells, are glucose hungry. But the other normal cells are oxygen hungry. It means that even in the presence of ample oxygen, ample molecules of oxygen in the environment of tumor, but they prefer the way of glucose using and the way of glycolysis. And this pathway of glycolysis is called Warburg effect. So now you know that Warburg effect is one of the important features of tumoral cells, and also some normal cells that are rapidly dividing. Some rapidly dividing normal cells. For example, lymphoid cells, normal lymphoid cells are very rapidly dividing. For example, in infections or in inflammation, the normal state of the body need rapid dividing of the cells. So these cells also use the Warburg effect. Okay, I said that tumor and the malignant cells are glucose hungry. By this feature, we can find them by some way of scanning, some imaging ways. Okay, we have a type of imaging, we call it PET scanning or positron emission tomography. We use from this way for finding the exact site of tumor, because tumor is very glucose hungry, and if we use from some types of glucose, for example, if we inject an exact type of glucose to a patient, the area of malignancy, for example, the primary site of tumor and also the metastatic site, show high level of uptake and high level of absorbance of this type of glucose, and we can find the site of them by this way of imaging, by PET scanning. So from this picture we can use in PET scanning, and patients are injected by a type of glucose that especially is fluorodeoxyglucose. This type of glucose taken up into tumoral cells and also to the normal rapidly dividing cells. And we know that most tumors are PET positive. As I said before, most tumors are PET positive, and also some other normal area are PET positive. For example, when we use from this way, sometimes we see, for example, an area of tumor, for example, in the liver, in a liver parenchyma, we can see the uptake of this glucose, and we say that this area is the tumor site, for example, primary tumor site. And sometimes we can see that some lymph nodes or tonsils are also positive. And we say that this area maybe are positive because this area is full of lymphocytes, and lymphocytes are normal cells that are rapidly dividing and they have high level of glucose uptake. Okay, as I said before, these two pathways of producing energy in the body, glycolysis and aerobic phosphorylation or mitochondrial phosphorylation. And the question is, why is it advantageous for a cancer cell to rely on this inefficient glycolysis? Do you know anything about this? You know why?
Because
glycolysis is like glycogen dividing to glucose, and the cancer wants to eat that glucose. So we have more glucose. And mitochondrial phosphorylation doesn't give us that much.
is that related to MYC gene, chromosome 8?
No. This is because of producing some intermediate metabolite in the pathway of glycolysis. In the normal cells and in a normal glycolysis pathway, we have producing of many metabolites like nucleic acid, proteins, lipids, and many other things that the cells need for dividing and for proliferation. Okay, because in this way we have many of this metabolite, of this intermediate metabolite. Because of that, because the cells for rapid dividing and for proliferation need this organelles, need this metabolic, they use this pathway for proliferation. And another important point is, as I said before, that rapidly proliferating healthy cells rely on aerobic glycolysis, and Warburg effect is not cancer specific. Okay, and you know that a growing cell has many requirements like nucleic acid for DNA, RNA, protein, lipids, and any organelles. So the answer is here. While pure oxidative phosphorylation yield abundant ATP, it fails to produce any carbon component that can be used to build the cellular component needed for growth, like protein, lipid, and nucleic acid. So the answer is simple. Aerobic glycolysis provides rapidly dividing tumor cells with metabolic intermediate that are needed for synthesis of cellular components, whereas mitochondrial oxidative phosphorylation doesn't. So, now the question is, how is this reprogramming of metabolism or Warburg effect triggered in the proliferating normal and malignant cells, and how this reprogramming happens in the cells? Any questions in neoplasia and in the malignant cells? Answer sometimes is mutations. we have many mutations in the normal pathways of cells in the malignant cells. The metabolic reprogramming is produced by signaling cascade downstream of growth factor receptor, the very same pathway that are deregulated by mutations in oncogen and tumor suppressor genes in cancers. So, whereas aerobic glycolysis ceases in normal cells when they are no longer growing, but in cancer cells, this reprogramming persists due to the action of oncogenes and loss of tumor suppressor gene function. Do you understand this slide?
Yes.
Who can explain? Can you explain?
It's saying that the cancer are related to mutation most of the time, and this mutation are using the aerobic glycolysis. As we said, the aerobic glycolysis is varying metabolism.
Intermediate metabolites. They prefer intermediate metabolites, you say.
Yeah, unlike the mitochondria pathway, which don't make intermediate products.
Okay, and this is because of, as we said before, due to action of oncogenes and loss of tumor suppressor genes function. Okay. Who can explain this picture? Okay, you have 3-minute time, okay, for study this picture, and after that, one of you come here and explain this picture, okay?
Oh, done. Do you want to take some minutes?
Okay, wait for our student. Done?
Yes.
You can come here,
Go.
except you. Okay.
Normal growth of the tumor cell, we have 3 mutations. One of the mutation can cause for VAS. VAS anyway can activate PK. PK will directly activate.
Activate.
Yes, activated PK.
No, this red line is for inactivation of pyruvate kinase.
Oh.
And the
The
green
green
line,
line
the
is
green
for
line is
activation.
yes, for activation and for upregulation.
Hope does mean mutation there will cause an inhibition of
Yes.
conversion of the phosphor.
PK is
Phospho
pyruvate
non-pyruvate.
kinase enzyme. Pyruvate kinase, yes, pyruvate kinase enzyme.
So we stop the conversion to pyruvates in a way that will cause the increase in nucleic amide.
Yes, exactly.
synthesis.
The inhibition
Will
of
cause the
this
growth.
enzyme, and also upregulation of glucose transporter can increase in the upstream area of this pathway. So when we have inhibition of pyruvate kinase and upregulation of glucose transporter, we have many nucleotide formation and many amino acid formation that they are need for growth.
And the mutation in PI3K will cause the increase in AKT, in way will cause protein synthesis and will cause the growth.
Yes, exactly.
Or mutation there will cause also the upregulate of the glucose. That means more molecule for glucose will enter. Anyway, will cause more glycogenesis. At the same time, more glycogenesis and more amino acid synthesis. Right?
Yes, yes, exactly. Thank you. Thank you so much. Okay, in this pathway, you can see that in a normal cell, this pathway is for normal, and this area is for abnormal and malignant cell. Okay, this pathway is for glycolysis. And we have growth factor receptors in any cells, and with the growth factors, we have some signals to the nucleus in one hand, and also we have some other signals to this metabolic pathway, other signals to the glycolysis pathway. Okay, these signals is inhibitory for pyruvate kinase, and in the other hand, this pathway by AKT mutations can upregulate the glucose transporter and can increase the uptake of glucose in the mutated and cancerous cells. By these mutations, by all of these mutations, finally we have increase in production of nucleotide amino acid protein that they are need for the growth. Any question?
So what's the name of the chapter? The name of the chapter.
The name of chapter. This chapter is neoplasia, and now we're talking about the hallmarks of cancers. Yes. If you want to find hallmarks of cancers and altered cellular metabolism. Okay. Now here you can see that we have, as we said in the previous picture, growth factor receptor signaling. In one hand, send some signals to the nucleus for proliferation and for dividing, and in the other hand, they also can influence the metabolism by upregulating glucose uptake and inhibiting the activity of some enzyme like pyruvate kinase, and finally increase in upstream glycolytic intermediate, as we said, amino acids, nucleotide, protein, and other things. And the other important thing was about RAS signaling. As you see in the picture, the RAS signaling finally along the PI3K and ACT pathway can upregulate the expression and activity of glucose transporter, and they can increase the glycolysis, and after that lipid biosynthesis and stimulate the protein synthesis. And also in that picture, a line was for MIC, MIC mutation is very important, other mutation in the malignant cells, and the pro-growth pathway upregulate expression of transcription factor MYC, which drives changes in gene expression, some changes in gene expression that support these types of metabolism. Okay. By contrast, many tumor suppressors can inhibit metabolic pathway that support growth. We know that we have some oncogenic factors, and we have some suppressor genes, okay, and they are in a balance in the normal cells. They are in a delicate balance for normal proliferation and normal apoptosis. We have already discussed about braking effects on the tumor suppressors. We have other tumor suppressor genes like NF1 and PTEN on signals downstream of growth factor receptors and RAS that allowing them to oppose the Warburg effect. You know that P53, that is most important tumor suppressor genes, can inhibit the expression of many genes that are involved in synthesis of cellular building block. Okay, p53 is a tumor suppressor gene, and it can inhibit the expression of the genes that are used for synthesis of this metabolite, this intermediate metabolite. Okay, there are two other links between metabolism and cancer. One of them is autophagia, and the other one is about oncometabolites. What's the meaning of autophagia?
Automatic cell.
Auto, what's the meaning of auto?
The cell digests itself. The cell digests itself.
Yes, yes. Why this happen?
I told you.
Understand.
Why does this happen?
Because
And when, for example, when this happen? Autophagia.
Mutation in the lysosome.
Infections.
No, apoptosis is different.
Infections, maybe.
No. Maybe sometime in infection, inflammation, any situation that nutrients are decreased in the environment and cell have no nutrient for using and for producing energy, for producing ATP, they start to consume and eating their organelles. Autophagia is a state of severe nutrient deficiency. Cells, not only in this situation, arrest growth, and also they start to consume their own organelles like protein, membrane, and any carbon source for energy production. If this adaptation fails, the cells die. Okay. Tumoral cells often seems to be able to grow under severe nutrient deficiency. Okay, the tumoral cells are able to live in this situation, in very bad situations. Okay, and they don't trigger and don't start the process of autophagia, and maybe this is because of that in autophagia, the autophagia pathway is disrupted in the tumoral cells. But now we know and we understand that tumoral cells sometime and also some tumors can trigger this pathway, can start the autophagia. And this is very important and is responsible for the resistance of some tumors to the therapies, to the chemotherapy and radiotherapy, you know? Because the chemotherapy and radiotherapy and many therapy that we have now for treating the malignancy can kill the tumoral cells that are active, that are actively dividing. Okay, they can find this cell and can kill them. So when a tumoral cells went to a sleep phase, to a quiescent phase by autophagia, they can live for more time without killing by the therapy and by the drug. We know that genes that promote autophagia have tumor suppressive activity. And the question is whether autophagy is always bad for tumor or not. In the condition of severe nutrient deprivation, tumoral cells may use autophagia to become dormant. You know the meaning of dormant? Inactive, yes, like a sleeping phase. Yes, a state of metabolic hibernation. You know metabolic hibernation? This is again sleeping, like we say winter sleeping, okay? That allows cells to survive for a long period. Such cells are believed to be resistant to therapy that kill actively dividing cells. Okay, so this phenomenon can be responsible for drug resistance in the tumors. Now about oncometabolism. Another group of genetic alteration or mutation in the enzymes of Krebs cycle. A very important enzyme in this cycle is isocitrate dehydrogenase. This is very important. Please remember this enzyme because this is very important for oncogenesis, and mutation in this enzyme is one of the important relationship of tumor with oncometabolism and the oncometabolite. They have revealed a new mechanism of oncogenesis that term oncometabolism. Okay. Steps in oncogenic pathway involving IDH or isocitrate dehydrogenase. Isocitrate dehydrogenase acquire a mutation. After this mutation, they can produce a specific amino acid, and after that, the mutated protein loses its normal ability in the normal situation. And after that, they acquire a new enzymatic activity. So, a mutated isocitrate dehydrogenase have another activity, have a different activity from the normal isocitrate dehydrogenase. And finally, they can produce another protein that we call them 2-hydroxyglutarate or 2HG. And after that, these 2HG act as an inhibitor of other enzymes that are members of TET family, like TET2. This TET family is very important in normal methylation of DNA, in normal action of DNA. This protein is very important. So, loss of TET2 activity leads to abnormal pattern of DNA methylation. And finally, all of them can cause expression of cancerous genes, and finally the cancerous cells and malignance. So isocitrate dehydrogenase can produce 2HG or 2-hydroxyglutarate, and after that this can inhibit the normal activity of TET2 on DNA methylation, and finally we have aberrant expression of the cancerous genes. Here in this picture, who can explain this? 2 minutes. You have 2 minutes for explaining this. Done?
Done.
Come here. No.
So,